Welcome to flow injection analysis and sequential injection. So in flow injection analysis, a liquid sample is injected into a continuously flowing liquid carrier that contains a reactant that reacts with our sample. So we have our carrier flowing in past our pump. Our sample is injected here at our injection valve, goes through the mixing coil, and then over here it goes through our flow cell. We've got some light path in, generally through a fiber optic cable. We've got our light path out going to our detector, and then our carrier plus sample goes to waste. So additional reagent can be added farther downstream from our injection valve, and the sample zone broadens as the sample travels from the injector here down to our detector. The sample also reacts with the reagent and forms some product, and then our detector is responding to that product. Now, flow injection analysis has some advantages over batch processes, which are separately analyzing individual samples. The first is speed, because often around 100 samples can be analyzed per hour. We also have automation of solution handling. So we have an auto injector that we can load up with a bunch of samples, and it will automatically run a whole series of samples, and we don't have to sit there and inject each one manually. It's also fairly low cost. Auto samplers can hold hundreds of samples, and this means that our labor cost is fairly low. We can set it up once at the beginning of the day and let it run for an hour or more as it runs through a whole bunch of samples for us. The figure here shows how the sample zone broadens and reacts as it flows through the tube. So when a liquid flows slowly through a tube, the flow is laminar, which means the flow at the walls of the tube is zero due to friction, and this causes the flow in the center of the tube to be faster, which means that in the center of the tube now, our sample is moving faster than it does at the edges, and we get this curved region here at the leading edge, and we get this curved region at the trailing edge, where in the center our sample is moving faster and our carrier is also moving faster at the trailing edge there. Now, liquid near the walls is mixing with the bulk via diffusion along the tube radius, so it's mixing along that direction. This radial mixing or radial diffusion gets faster as the radius of the tube gets smaller, and at typical tube diameters of 0.5 to 0.75 millimeters, then the radial diffusion is significant in just a few seconds. The sharp bends in and the curvature of our tube creates turbulence, which just further enhances the mixing. And as our sample band moves through the mixing coil, then the reaction between the sample and the reagent can only occur where they mix, which is at the leading and trailing edges. So we have reaction occurring here at the interface between our sample and our carrier with our reagent in it, and at this interface back here between the carrier and our sample. And so as our sample progresses through, the zone, that sample zone is broadening, and that reaction can only occur in those regions. And so now the darker region here, the darker blue there and here, is representing our sample. The lighter blue region here and there is representing the zones which are rich in product, and we don't have as much product in the center because we don't have any of our reagent to react with our sample there. Now, the formation of product depends upon the rate of mixing and the reaction rate. And because the time between injection and detection is typically under a minute, then the mixing of sample and reagent is incomplete, and equilibrium has not been attained before detection. So now let's talk about sequential injection. This uses flow programming and flow reversal controlled by a computer. And note here that flow is not continuous as it was in flow injection analysis. So some advantages over flow injection analysis include smaller reagent volumes, less waste is generated, and sequential injection can be used for constant monitoring of environmental or industrial processes. Another advantage is that the instrumentation has been miniaturized to what we call lab on a valve, So we have a relatively small instrument here, and the key is the 6-wave valve in the center here. This allows the computer to control whether the sample or reagent is being allowed to flow. The syringe pump over here can draw liquid, either sample, reagent, or carrier buffer. The carrier buffer is accessed via the 3-way valve up here. That syringe pump can draw our liquid into the syringe or expel it out of the syringe and into the holding coil. The holding coil is up here and is temperature controlled. So here's an example experiment. Our sample is drawn via port 5 into our holding coil. So we draw the sample in. Then reagent 1 is drawn via port 3 into the holding coil. Flow is stopped for a while to allow mixing and the reaction to progress. So that's here. We've drawn our sample in this phase. Then now we're drawing in our reagent, and we're allowing them to mix here. We stop the flow, and we allow them to mix, and we get our product forming there in the lighter blue band. And now flow can be reversed to send liquid out port 2 and through our flow cell. So we have then the syringe pump that liquid back out of our holding coil, and we can then measure the absorbance at some wavelength of interest. Now, additional reagents could be added, or volumes or reaction times could be changed. The flow could be stopped when the product zone reaches the detector to observe additional product formation if there's more being formed. We could stop our product there and do some sample averaging if our signal is especially low. So that was flow injection analysis and sequential injection.